Good afternoon, students. Here again, I am present with the new topic, that is the seed viability, and this is the same topic which will be coming under the reproduction in flowering plants, and this is the topic for class 12th CBSE board biology. So here we will see that what we will be meaning about seed viability. What is the meaning of seed viability? So let us see over here that whatever the seed viability is there, whatever the seed viability is there, we can see that it is the period of time for which the seed retains the ability to germinate. It is the period or time for which the seed is able to retain its germination. Okay, so let us see it again, that it is a period of time in which the seed retains the ability to germinate. The seed retains its ability to germinate means for how much long a seed can be kept, and after which again if it is sown, then how it can grow into a new plant. So this is called as seed viability. The seed viability is determined by genetic as well as environmental factors. As you have seen that in the previous segment, that is the viability of the pollen grain, that the genetic and the environmental factors, they were responsible for the seed viability. So here we can say that whatever the seed viability is there, the seed viability is also depending upon the genetic factor as well as the environmental factor. So now what environmental factor can be there? The environmental factor may be the humidity or the temperature. Means if the humidity or temperature we are considering, these environmental factors, they are responsible for the seed viability. Okay, so the seed viability can be depending upon the humidity and temperature. These are our environmental conditions. So now on the genetic basis, let us see that the seed viability ranges from few days. That is from 1 season to 2 to 5 years, and sometimes it is also lasting for 100 years. Okay, so 2 to 5 years in most of the plant crops, and 100 years, example for trifolium. Okay, what is trifolium? Whatever the balsam is there na, balsam is a fodder plant. It's a fodder plant which is used for the cattles. So this trifolium can have its seed viability for about 100 years. So means its seed can live for 100 years. Trifolium seeds can live for 100 years. Now the seed viability has been found out to be more than 1000 years in lotus. So we can say that the seed viability of lotus is for 1000 years. Even its pollen grain is also living for a long time. Its pollen viability is also very much, and its seed viability is also for a longer period of time. So now we can see here is 2000-year-old a Phoenix dactylifera. They were excavated from the King Herod's palace near the Dead Sea. There is a place, Dead Sea. There a king's palace was there. King Herod's palace was there, and in this palace some grave was found. In this grave some Phoenix dactylifera seeds were there. These are the date palm seeds, children. Actually it's a date palm variety. In Hindi we can also call it as khajur. So this variety, it can have its seed viability for almost 2000 years. So it's quite a long period of time. For 2000 years we cannot imagine that any seed can live, but though it's a reality that Phoenix dactylifera is having its seed viability for such a long time. Okay, similarly 10,000 years old seeds of lupins. Okay, lupins, it's a type of plant. Lupin is a type of plant which was also excavated from the Arctic tundra. Tundra means a very colder place. The north and the south poles, the north pole or the south pole, they're called as the very cold places. So if you talk about north, that is Arctic tundra. Arctic tundra is having one of the variety of the plant called as lupin. This lupin seeds, they can live for almost 10,000 years. It is breaking the record of even this lotus, as well as it is breaking the record of Dactylifera. So we can see that these are the plants which have large number of the or large period or a huge period of seed viability. So we can say that its seed viability is very much. So let us see one more time that what is seed viability. See, it is the period of time for which the seeds retain the ability to germinate. Very, very important. This heading you have to learn that whatever the ability of the seed by which it retains its germination power, the ability of the seed by which it retains its germination power is known as its seed viability. And the factors which are responsible for that are environmental factors as well as genetic factors. Okay. Now let us see the viability of the seed. The viability of the seed is tested by its, means how you can check the viability of the seed. See, if the seed you sow, either you sow or you soak in water, either you sow the seed or you soak the seed in water and it starts respiration. If you are soaking the seed in water and it starts respiration, then you can say that the seed is viable. Means the inside the endosperm of the seed, if the embryo is living, if the embryo is still living, then it will start respiration. Okay, it will start respiration, it will obtain energy, and then what it will do, it will start germination. After the respiration, it will obtain energy, and with that energy, with the help of that energy, it will start germination. Okay, so now let us see that respiration or the respiration in the seeds, how you can identify. So you have to use a reagent, you have to use a chemical. The chemical name is triphenyl tetrazolium chloride. This triphenyl tetrazolium chloride, which is colorless, this colorless triphenyl tetrazolium chloride, it will turn pink. Why it will turn pink? When it will get the CO2. And how it will get CO2? When it will start respiration. When the seed will start respiration, it will release CO2. And what will happen? That whatever the triphenyl tetrazolium chloride is there, it will turn triphyl or triphenyl formazine, and it's a pink color compound. So you can check, you can test that yes, a seed is doing respiration, and by the help of respiration only, seed will start germination. Okay, you can see in this diagram that the light energy is required, and during the light energy, what will happen? Chloroplast will undergo photosynthesis and it will form the food, and this food which is stored in the cells will be respired into the mitochondria, and by the help of mitochondria, in the mitochondria the respiration will occur and it will provide energy. Okay, now next we should see that now the next topic is apomyxis. What do you mean by apomyxis? See, apomyxis means Apomixis means without marriage, means without any sexual reproduction. If the seeds are formed, then it is called as apomixis. What is apomixis? It is the formation of new individuals by asexual methods which mimics the sexual reproduction, including seed formation, but do not involve any gamete fusion. Means in this, what is happening? Any gamete fusion is not required. Any gamete fusion means either the egg cell or the pollens, they are not required. It is just the copy, it is just the mimicry of the sexual reproduction, though it's a asexual reproduction. Means it is the formation of new individual without any sexual reproduction. In this, what is happening? Only the asexual method is applied. Asexual method is applied, and this asexual method will be forming new individual. Asexual method is forming new individual. So what is apomixis? The formation of new individual by the help of asexual method, and it is just the mimicry. It is just the mimicry of the sexual reproduction. In this, what is happening? Seed formation is also done without any involvement of the gamete fusion. Okay, hope you people are understanding. Now the next is normal type of sexual reproduction having two regular features. In sexual reproduction, what is having? What features are there? The first feature is meiosis, and the next feature is fertilization. You know that the gametes are formed by meiosis. The gametes are formed by meiosis, and next what is happening? After the gametes are formed, these gametes needs to be fused. These gametes needs to be fused. Okay. So what is happening? That the meiosis is occurring, by which gametes are formed, and then fertilization is occurring, by which fusion has to be done of these gametes. Okay, the male and female gametes, they have to be fused, and then the fusion will be occurring. So this is known as epimixis. Okay, the normal sexual reproduction is also called as epimixis. Okay, the normal sexual reproduction in the plants is called as epimixis, whereas the asexual method, without the involvement of the sexual method, it is just the mimicry of the sexual method, it is called as apomixis. So did you understood the difference between apomixis and epimixis? So there are two differences between both of them, so which you have to learn. Now the next is the organism producing through apomixis is called as apomix. The organism producing by the help of apomixis, the organism by reproducing by the help of apomixis is called as apomix. The organism reproducing through apomixis process is called as apomix. Next, apomixis is controlled by gene, and individuals are genetically similar to the parent producing by them. So it's a type of an asexual method. So you know that in the asexual method, clones are formed. So similar, what are clones? Clones are the individuals which are similar to their parents. What are clones? Which are similar to their parents, So in apomixis, in this type of reproduction, in apomixis, which is an asexual form of reproduction, clones will be formed. The exactly similar individuals, the exactly similar offsprings will be formed. They will be looking like their parents. Okay. Now, whatever the apomixis is there, it is occurring by the following methods. The apomixis occurs by the following methods. What are these methods? See, the formation of asexual seeds. If the embryo develops directly without gametic fusion. So you know that what is apomixis? The asexual reproduction, which is just the mimicry of the sexual reproduction. Okay, in this exactly the gametic fusion, the pollen grain or the male gamete as well as the female gamete fusion does not occurs. But then also the embryo formation will be there, the embryo development will be there, the zygote formation will be there. Okay, the zygote is formed, and this zygote will be developing into an embryo afterwards. The second one is sporophytic budding. You know, the spores will be formed, okay, and the spores will be undergoing budding. And if embryo develops advantageously from diploid cells of nucellus or integument, okay, in this what is happening? Sporophytic budding. Spores will be formed and these spores will undergo budding. Spores will be formed, spores will undergo budding, okay, and these buds which are sideways, which are adventitious, lateral, sideways, they will be forming diploid cells. They are actually 2n cells, diploid cells, okay. And like this also, by the sporophytic budding, apomixis can occur, okay. Hope you people are understanding. Now let us see the next topic, parthenogenesis. See, let us see this word, let us break it and understand. Parthenos means virgin and genesis means formation or descent, virgin descent. So what it is having, the literal meaning of, or the literary meaning of it is the development of a new individual. What? Development of new individual from a single gamete without the fusion of another gamete. Means a single gamete, one type of gamete is forming a new individual. Only one gamete is forming a new individual. Means the fusion of the gamete with the another gamete is not occurring. The male and female gamete will not fuse together. Only one gamete will be forming the individual. This is called as parthenogenesis. So what is parthenogenesis? Parthenogenesis is the process in which a new individual is formed by single gamete. When one gamete, either the male gamete or the female gamete, it is forming a new individual, and the fusion of gamete is not occurring. The male and female gametes are not fusing together. Only one type of gamete is forming a new individual. That is called as parthenogenesis. Okay. So now, depending upon the ploidy of the gametes, there are two types of gametes. Depending upon the ploidy of the gametes, there are two types of the gametes. They may be haploid or diploid gametes. Means how the chromosomes of the gametes are divided, based upon them the gametes can be also divided into further two types, like haploid gametes or diploid gametes. The haploid gametes or the diploid gametes. Okay, what are haploid gametes? Which have half number of chromosomes. And what are diploid gametes? Which have 2n, that is the pairs of chromosomes. Okay, this is haploid, this is diploid, which you know very well. Now in haploid parthenogenesis, when the gametes are haploid, then the haploid parthenogenesis will occur. And what will happen? The embryo sac, you know, embryo sac, this is an ovule. Suppose inside this is an embryo, this is an endosperm. This endosperm is also called as embryo sac. Okay, in embryo sac, in haploid parthenogenesis, the embryo sac and its eggs are haploid. Means suppose if you talk about, in spite of directly talking about embryo sac, let us talk about ovule. Suppose this is the egg cell. So if this egg cell is haploid, okay, then it will undergo haploid parthenogenesis. If the egg cell in the embryo sac, if within the embryo sac the egg cell is haploid, means it is having half number of chromosomes. Then it will be referred to as haploid parthenogenesis. It will undergo haploid parthenogenesis. Okay. Now let us talk about diploid parthenogenesis. What is diploid parthenogenesis? When the embryo sac, when the ovule is going to contain egg which is diploid. Suppose this is the ovule and here the egg cell is there. Inside this the egg cell is there. You may also know na that suppose this is the ovule, these are antipodal cells, these are synergids, this is the egg cell, these are the central nuclei. Okay, so whatever the egg cell is there, this egg cell is there. If this egg cell is having diploid nature, though it should not be, but sometimes it is sometimes diploid. So if it is having diploid nature, this egg cell is having diploid nature, so this egg cell will undergo parthenogenesis and it will form a diploid embryo. This is called as ploidy. Whether it is forming the haploid embryo, whether it is forming the diploid embryo, it is called as ploidy. Okay, so now diploid parthenogenesis is generally accompanied by failure of meiosis during the megasporogenesis as well as direct formation of embryo sac. Okay, so diploid parthenogenesis is generally accompanied, the diploid parthenogenesis is accompanied by failure of meiosis. See, it is definite if meiosis will occur, why the diploid condition will be there if meiosis will occur. So meiosis fails. When the meiosis fails, then the diploid parthenogenesis will occur. And what it will form? It will form an embryo sac, directly an embryo sac from a nuclear cell. You can correlate from the previous topics which I taught you. So from the nuclear cell, if the embryo sac is formed, okay, if the embryo sac is formed from the nuclear cell without the megasporogenesis, then it is called as diploid parthenogenesis. Megasporogenesis occurs by meiosis. So if meiosis is not only occurring, meiosis is not going to have, then what will be happening? Then mitosis will occur, and at the same time what will be having? The diploid parthenogenesis will undergo. So let us again conclude that diploid parthenogenesis is generally accompanied by the failure of meiosis. Diploid parthenogenesis is generally occurring when the failure of meiosis is there, and the failure of megasporogenesis is also there, which will lead to the direct formation of embryo sac from the nuclear sac. It is taking place in the plants like poa, apple and rubus. Okay, now next topic is apogamy. So now let us talk about apogamy. Apogamy means without gametes, without gamos, without marriage, without any sexual reproduction, without any fusion. So it is the formation of sporophyte or embryo. It is the formation of embryo directly from cells of gametophyte. Means either the egg cell or the pollen cell, any of the gametophytes are there. If directly from them the embryo is formed, then it will be called as apogamy. Only diploid apogamy is successful, that is the gametophytic cell forming the sporophyte is diploid. Means when the sporophytic cell is diploid, then the diploid apogamy will be taking place. Okay, now let us talk about polyembryony. See, very important as per the exam view. So polyembryony, the phenomena of having more than one embryo. Quite interesting that when there are one embryo, at that time, if more than one embryo is occurring, if more than one embryo is formed, that is called as polyembryony. Okay, poly means many. So when many embryos are formed at the same time, it is called as polyembryony. Okay, it is called as polyembryony. So when more than one embryo is formed at a time, it is called as polyembryony. So occurrence of polyembryony due to fertilization or more than one egg cell, due to fertilization or more than one egg cell, is called as simple polyembryony. So what is simple polyembryony? What is simple polyembryony? Occurrence of polyembryony due to fertilization of more than one egg cell. So more than one egg cells are there. If more than one egg cells are there, then definitely it will form more than one embryo. If one egg cell is there, this one egg cell will be fertilized and one embryo will be formed. But if more than one egg cell is there, then if suppose two egg cells are there, then what will happen? These two egg cells will be fertilized and they will form two embryos. Okay, now let us talk about simple embryony again. That occurrence of polyembryony due to fertilization of more than one egg cell is called as simple polyembryony. Hope it is clear. Now next is formation of additional embryos can also occur from different parts of ovule. Sometimes the egg cell is leading to the formation of many embryos or the embryo, but there are many other parts also. Sometimes many other parts of the ovule which can form the embryos. They can be antipodal cells, they can be synergids, they can be nucellus, they can be integuments. So we can say that the formation of additional embryos can also occur from different parts of ovule, like synergids, antipodal cells, nucellus or integuments. When these kind of the parts, they sometimes get fertilized, they actually, when in these cells the exchange of the or the fusion of the gametes, the fusion of the nucleus takes place, then they may emerge as an embryo, and this will lead to the polyembryony. Okay, now these plants can be which undergo polyembryonic conditions. There can be citrus plants like oranges, groundnut can have this, onion, opuntia, mangifera you know very well, I don't need to tell you again and again, mangifera. Now who saw this polyembryonic situations? Leeuwenhoek, Antony van Leeuwenhoek, he discovered the polyembryony in the year 1719. He saw this. The same was confirmed by Schnarf. This is also the person, Schnarf, in 1929. He confirmed this. Suppose one person says anything, another person may not understand, but more than one person says that something, then sometimes it has to be considered. Now the polyembryony is most commonly seen in gymnosperms than in angiosperms. Means the gymnosperms show it more frequently as compared to the angiosperms. And polyembryony can be false or true embryony. Means sometimes it may seem that it will form a seed, but sometimes it may be false. Sometimes when it is forming seed, it may be called as true. Okay, so in false embryony, multiple embryos arise in different embryo sacs in the ovule, whereas in true embryony multiple embryos are formed in the same embryo sac. Means when within the same embryo sac, within the same ovule, you can say, what is happening when within the same embryo sac, same ovule, many embryos are formed, it is called as multiple embryony. Hope it is clear to everybody. Now the causes of polyembryony, causes, very very important. See, cleavage of proembryo. Suppose this is the ovule, here the embryo is formed. Suppose the embryo is formed. Now if this embryo gets cleavage, means it's dividing, then what happens? The same embryo will form many embryos. It is happening actually in orchidaceae. What is orchidaceae? Orchid plants. Sometimes the polyembryony happens or occurs in orchidaceae. Now, the development of many embryos from other cells of embryo sac except egg. We understood now that sometimes the integuments, the antipodal cells, the synergids, they fuse with the male gamete. And what happens? They form multiple embryos. Hope you people are understanding. Next is formation of many embryos, sometimes due to the presence of more than one embryo sac. So sometimes more than one embryo sac is present, due to that also polyembryony occurs. Now next is what? Formation of many embryos from structure outside the embryo sac. We can say there are many other structures, as we can see that integuments, these are the structures outside the embryo sac. Sometimes they also lead to the formation of polyembryo. Like in mango and in opuntia. Now the polyembryony is important for practical reason, because genetically uniform type of seedlings can be obtained. Suppose you want to obtain genetically same seedlings, same type of plants you want to get, then the polyembryony is playing an important role. Now, nucellar embryos. Nucellis word you understand. Nucellar embryos are qualitatively superior. To those obtained by vegetative propagation. So, whatever these nucellar embryos are there, by the polyembryony, whatever the plants are formed, they are sometimes superior than the vegetative propagation plants. Like two plants you form, one is formed by vegetative propagation, another is formed by polyembryony. Then this polyembryonic plant will be more better as compared to the vegetatively propagated plant, as it may have more diseases or something like that. Okay. So what we can say here, that nucellar embryos are qualitatively superior to those of vegetative propagation, as nucellar embryo seedlings are free from disease and maintain their superiority for long time. Means whatever the embryos are there, which are nucellar embryos, they will be lasting for a long time. They can survive for a long time in the harsh environmental conditions. Okay. Now next topic will be ours, parthenocarpy. What is parthenocarpy? Let us see. Parthenocarpy. Parthenos means virgin and carpos means fruits. Means the fruits which are formed without fertilization of the gametes. Though the ovary enlarges and becomes a fruit. The ovary enlarges and becomes a fruit after the fertilization. But suppose some instances are done in which without the fertilization the ovary enlarges, then what will happen? That without the fertilization, if the ovary enlarges and becomes a fruit, it is called as parthenocarpy. What is parthenocarpy? When the fruits are formed without any actual fertilization. So what is good about it? This produces seedless fruits. The parthenocarpy produces seedless fruits. Parthenocarpic fruits are seedless. As you can see here in this diagram, you can see this is the tomato which is formed due to fertilization of the ovary, and this is the tomato which is formed by parthenocarpy. Here you can see no seeds. Here you can see seeds are there. Okay, we can say here that here seeds are there, here no seeds are there. So many times we like the fruits which are having no seed. Sometimes we like this fruits which are having no seeds. Okay, so the fruits that have seeds with an asexual embryo or pseudo seeds are also parthenocarpic. Sometimes we also get pseudo seeds, though they are not actual seeds. Sometimes we also get pseudo seeds, though they are not actual seeds, but they look like seeds. In parthenocarpic plants, sometimes the pseudo seeds are also formed. Now let us see the type of the parthenocarpy. The parthenocarpy is of three types: the genetic parthenocarpy, the environmental parthenocarpy, and chemically induced parthenocarpy, which is very frequent nowadays, which is very common nowadays. Now the genetic parthenocarpy. See the genetic parthenocarpy, let us talk about. See, the parthenocarpy is due to genetic alterations caused by mutation or hybridization. It is also called as natural parthenocarpy. Okay, so when the parthenocarpy comes in a plant, it occurs in a plant, it comes in a plant where the mutation has been made, where the mutation has been done. Okay, where the mutation has been done, this mutation has arisen, and this mutation can be due to hybridization. Okay, then what it can be called as? It can be called as genetic parthenocarpy. Means when the fusion of two different variety gametes of the same species plant is done, that is called as hybridization, and due to that the parthenocarpy arises. This is called as genetic parthenocarpy, commonly called as natural parthenocarpy. Actually, in nature sometimes it occurs. Due to mutation in nature, it sometimes occurs. So, in apples, banana, pineapple, and the variety of grapes and pear, this genetic parthenocarpy is occurring. Now, the next one is the environmental parthenocarpy. See, the low temperature, forest and fog can induce parthenocarpy in number of the plants. Sometimes human are not doing this parthenocarpy. No human involvement. So at that time, what is happening? The environmental factors, they lead to the parthenocarpy. Means they lead to the formation of the fruit without even the fertilization of the egg cell. No fertilization. Without fertilization, when the fruit is formed, that is called as parthenocarpy. Without fertilization, when the fruit is formed, which is called as parthenocarpy. And if parthenocarpy is induced, if it is done by environmental situations like low temperature, frost, fog, etc., it is called as environmental parthenocarpy. Okay, children, hope you people are understanding. Now, chemically induced parthenocarpy, very very important. Nowadays it is happening very commonly. So what is happening? A spray or paste of auxin and gibberellin is made to get sprayed. A spray of paste of auxin and gibberellin in low concentration, that is 10⁶ or 10⁷ moles, has been found to induce parthenocarpy in some plants. Means when auxin and gibberellin hormone are sprayed on some plants like tomatoes, lemons, oranges, and strawberry, as well as the blackberry or the figs, then it is called as chemically induced parthenocarpy. Okay, now the thing is, the parthenocarpy is very understood to you. You have understood, no confusion in this. Now let us talk about a true fruit. See, a true fruit develops from the ovary, and the false fruit may develop from floral parts, that is from thalamus, and it may contribute to fruit formation. The thalamus may contribute to the fruit formation. Means the true fruit is formed from ovary, and the false fruit may be developing from other parts or other than ovary. Now what is parthenocarpy? The fruits develop without fertilization. Example is banana. Now what is the importance of the parthenocarpy? See, the parthenocarpic fruits, they do not contain seeds. So there is no need to remove the seeds before eating. So there is no need to removal of the seeds. So these fruits we will be considering as at a great or the high cost, because they don't have seeds. Sometimes we do not require seeds for a normal eating purpose. Sometimes we do not sow the seeds, so we don't require the seeds, we only want fruit. So at that time the parthenocarpic fruit is very important, or it is good. Now the fruits can be developed inside the greenhouses where pollinators are not available. So parthenocarpic fruits are developed in the artificial situations. Okay, these are developed in the artificial situations where we do not require any kind of a pollinating agent, okay. There we can go for the parthenocarpy. So parthenocarpy is helpful producing the fruits in the greenhouses. The parthenocarpy is able to produce the fruits in the greenhouses, or it leads to the formation of fruits without fertilization of the ovary. Okay, hope you people are understanding and you people are safe. You may be continuing to wash your hands. You are continuing to wash your hands with the soap, or you may be rubbing your hands with the alcoholic sanitizer, and you are using any tissue while you are coughing and you are discarding that tissue into the dustbin, as well as you may be using a handkerchief which you may be washing after a fixed regular interval of time, and you are avoiding to touch your face. And you are also avoiding to touch anything with your right hand. Avoid touching it with the right hand. Touch the things, the external things, that is the doorknobs or the gate, with the left hand, so that if you are touching the things or touching your face with the right hand, you may avoid any contamination. Next is, we will be meeting again with a new topic. Till then, thank you.